It is given by Dr. Amin Saberi. Dr. Saberi earned a medical doctorate from Iran and is currently a PhD student at the Cognitive Neurogenetics Lab at the Max Planck Institute for Human Cognitive and Brain Sciences and Research Center Jülich. And today he will be talking about laminar thickness covariance in the big brain in association to cortical hierarchy and connectivity. So the floor is yours.
Thank you very much for the kind introduction and the opportunity to present my work here. Okay, so I hope you're seeing my screen.
Yes.
Okay, yeah, thank you very much. Okay, yeah, so I'm going a bit into a different direction. I'm going to talk about histology, and specifically I'm going to talk about how laminar thickness in the cortex varies and what is its functional meanings for the cortical function. Okay, so we know that the cortex is a layered structure and it consists of 6 layers, and each of these layers have their own distinct features, and cytoarchitecture, myelin architecture also function. But we also know that this layered structure is not homogeneous across the entire cortex, and it varies a lot across different regions. And then characterizing this variation across different regions has been an important focus of the field over the past century. And here you can see 2 examples of this that are nearly 1 century apart that shows laminar structure variation across the cortex. And while this has been great and very helpful, the previous research, but they have been mostly based on theory, meaning that expert histologists, anatomists have looked at samples of tissue from cortex and then try to categorize them into different categories of laminar structure, as you can see here. And also, they have been based on a sample or data that has a low sampling rate, meaning that it doesn't cover the entirety of the cortex. Here, we decided, or we aim to revisit this century-old question from a new angle and then use a whole brain data or whole isocortical data, and also use a data-driven approach to avoid biases introduced by using a theory. And the data that we used was a ultra-high resolution histological atlas of a human brain of a 65-year-old male, which has been cell body stained and then sectioned at very high resolution at a resolution of 20 microns. And this is also available online if you were interested, look it up. And then previously, a colleague of mine has segmented the cortex of this brain, this atlas, into the 6 layers. And this was the main input data to our study. So we had the thickness of these layers, of the 6 layers, at each location of this particular cortex. And we asked what is the main pattern of variation in laminar thickness in this cortex. And then we were also hoping that it generalizes to other individuals as well. Okay, so then to do that, we first did some pre-processing on the data. First of all, we excluded some of the regions of the cortex, which are called agranular or disgranular regions, because these regions are supposed not are suggested not to have a 6-layer structure. So therefore, it was not appropriate to assume 6 layers in these areas, which are mostly limbic areas. We then smoothed the maps, and we did this mainly to get rid of the local effects of curvature on laminar thickness. So this is an effect known as Bok effect, which is as we move from sulfide to gyri, there is a thickening of deeper layers, as you can also see this here in our data. And then if we apply smoothing, as you can see, most of this effect is gone. And then we normalized the thickness or the absolute thickness of each layer by the total thickness of the cortex at each location to get the relative thicknesses or the proportion of how much each layer is present at each location. And then we apply the parcellation, ultimately getting a matrix like this that shows the relative thickness of layers 1 to 6 in each area of the cortex. Okay, so then we wanted to see which regions have similar laminar pattern or laminar structure pattern to other regions. And to do that, we took partial correlations of these 6 element arrays between different regions and calculated a matrix which is called laminar thickness covariance matrix, or LTC. And then to understand the structure behind this matrix, and also to get a map of overall variations in the laminar structure, we applied dimensionality reduction on this matrix, and specifically PCA, and focused on the main principal axis derived from this dimensionality reduction. And we called it LTC G1 short for principal gradient, and we called it gradient because it shows gradual variations. And this map, what it shows is basically that there is a shift from the dominance of more deeper layers 5 and 6, in frontal areas, towards the dominance of superficial layers 2 and 3 specifically, towards the more posterior regions in occipital and parietal lobes. Okay, so this was about laminar thickness and how it varies overall across the cortex. But then what about the other features of laminar structure? And specifically, one data that was available to us from this high-resolution atlas was the density, or it was image intensity, which reflects a sum of neuronal density and size in each area and in each layer. And then we observed that if we combine both features, thickness and density, and then apply the same approach, we get an axis which is very similar to the main axis that we observed, which shows that these two features co-vary with each other. And specifically, in this axis, we see that, again, from frontal to more posterior areas, we see the same pattern in thickness, but also an overall increase in the density of the image, which is more prominent in middle layers. Okay, so then after characterizing this data-driven map of laminar structure, laminar thickness variability, we asked what is its functional relevance. And one theory that relates laminar structure to function is that there has been several animal studies showing that regions with different laminar structure tend to have laminar projections to each other that can be predicted based on how the laminar structure is different between them. And specifically, we can predict whether these laminar projections are feed forward, feed back, or columnar. And then the pattern of these projections can be used to construct a cortical hierarchy. And then, therefore, we hypothesize that the variation of laminar structure may relate to cortical hierarchy. Okay, so then we used two maps of cortical hierarchy based on macaques and humans, and then studied its relation to the map of laminar structure variability that we found. So first, starting with the macaque cortical hierarchy map, this map is based on the very same, with the definition of, or it's called laminar-based hierarchy map, which is based on the feedback versus feed-forward pattern of connections. Generally, it assumes that regions that are higher up at the hierarchy tend to send more feedback connections, and regions that are lower tend to send more feed-forward connections. And this approach is only available in macaques because it's based on track tracing, which we cannot do in humans. But then also our map was based on humans, so we had to map it to the macaque cortex. And then when we looked at the association, we observed a relatively high correlation, overall showing that the infragranular dominant regions in more frontal areas tend to have higher hierarchical positions. Okay, so but what about humans? In humans, we cannot do track tracing, right? But we can do imaging. And then based on imaging, and specifically based on resting state functional MRI, we derive the effective connectivity matrix, or we obtained this matrix from another publication. And then asked whether there are regions that are more influencers on the other regions rather than being influenced by them. And this was the basis for another definition of hierarchy, which we call asymmetry-based hierarchy, which is based on asymmetry of effective connections between nodes. And generally, in this map, if regions that tend to send more, tend to have more stronger outgoing connections rather than incoming connections, would have higher hierarchical position. And again, we see that in this human asymmetry-based hierarchy map, we see a relatively high correlation with the main axis of laminar thickness covariation, again showing that the more infragranular dominant regions tend to have a higher hierarchy. Another functional relevance feature that was interesting to us to look at was the relation of laminar thickness covariance or laminar structure covariance to probability and strengths of connections. This has again been shown in several animal studies that as two regions become more and more similar in their cytoarchitecture and laminar structure, they're more likely to be connected, and their connections would be stronger. We wanted to test this, and we used the structural and functional connectivity matrices obtained from the HCP dataset, which is neuroimaging data, and not from the same individual, not from BigBrain. But then this asked whether a higher laminar thickness covariance, which shows higher similarity of laminar thickness patterns, would predict higher likelihood of structural connections or higher strengths of functional connections? And the answer was yes, although the effects were not very strong, but we observed that as two regions become more similar in their laminar thickness patterns, the likelihood of their structural connectivity and the strengths of their functional connectivity get stronger. But this effect was not there when we looked at long-range connections, which is interesting. So to sum up, we found we described a data-driven axis of laminar structure, laminar thickness variation from frontal areas more infragranular dominant to posterior areas more supragranular dominant. And we observed that this was also co-aligned with variations in laminar density. This variation in anatomy was aligned with the cortical hierarchy that we mapped based on laminar connections in macaques and effective connectivity in humans. And also we observed that higher likelihood and higher strengths of connectivity can be predicted based on the similarity of laminar structure between two regions. Thank you very much for your attention, and thanks to my co-authors and supervisors. And if you're interested to know more, this work has been recently published, and you can find it under this QR code. Thank you.
Thank you so much. That was a great talk. Oliver, do we have a question?
Yeah, we do have a couple of questions in the chat. So first, Marco Palombo asks if you have also more quantitative measures of cell body density per each layer, and if you have those data projected onto those surfaces, and if they have not, are you planning to compute them? If not, would they be able to work? Would they be open to work in a collaboration on that?
Yeah, thank you very much for the great question. Yeah, actually, yes, there is some data available that has. Look, maybe I can show my slides again. There is a slide on this, but it's hidden. Never mind. It's here. Oh no, sorry. Okay, this is it. I hope that you're seeing this one. So yes, this data of looking at individual cells is available, but not for the entire cortex of BigBrain, because BigBrain original resolution is 20 microns, and in that resolution you cannot see individual cells, and you cannot look at their density or size. But in very specific locations, this is a work in progress that I'm also not involved in, but they image this same brain at a much higher resolution, at a resolution of 1 micron. Therefore, you can see individual neurons, as you can see this in this example image. And then a colleague of mine has segmented these using again an AR method. And yes, we did look at this preliminary data and observed that this axis that we found does also correlate with some of the measures of neuronal density and neuronal size in different layers. Yeah.
Okay, thank you. Maybe another question from the chat. So could you talk about how your results about laminar thickness across layers could vary with age, sex, and ethnicity?
Yes, I would be very curious about that. As I mentioned, this is just one brain, right? So we cannot, we are not sure if this would generalize to other brains, or how it would, how there would be inter-individual differences. And I cannot really answer that until more brains are available. There is a BigBrain 2 coming soon, I think, which is a younger individual. I don't know, I think it's the same sex and ethnicity, but I'm not sure. But yes, I mean, it's just a matter of data availability, but that's a very interesting question that I'm also curious about.
All right, great. Thank you, Amin.